Pharmapal.
QSAR models and pancreatic lipase inhibitors. Hey everyone! Welcome back to our channel. Today, we're diving into some fascinating research on quantitative structure-activity relationship, QSAR, models and their application in discovering new pancreatic lipase inhibitors. So, grab your notebooks, and let's get started. First off, what exactly is a QSAR model? QSAR stands for Quantitative Structure-Activity Relationship. It's a computational method used to predict the activity of chemical compounds based on their molecular structure. In simple terms, it helps scientists understand how the structure of a molecule influences its biological activity. In this study, researchers collected data from existing literature to build QSAR models using the Monte Carlo method. This approach allows for the evaluation of many different molecular configurations to find the most effective ones. Now, let's talk about the results of the best QSAR model. The statistical parameters showed a correlation coefficient, R superscript 2, of 0.864 and a cross-validated coefficient, Q superscript 2, of 0.836 for the test set. For the validation set, these values were also impressive, R superscript 2 equals 0.824 and Q superscript 2 equals 0.819. These numbers indicate a strong predictive power of the model. Key descriptors influencing activity. The study identified several molecular features that positively impacted the activity of the inhibitors. Branching, the presence of branched structures in molecules. Oxygen with branching in double bond, specific arrangements of oxygen atoms. sp superscript 2 carbon, a type of carbon atom that is part of a double bond. Cyclic rings, rings in the molecular structure that can enhance stability and activity. Using the QSAR model, researchers designed novel pancreatic lipase (PL) inhibitors with improved predicted activities. They performed molecular docking simulations to see how these new compounds interacted with the active site of pancreatic lipase. Key interactions. The docking studies revealed that these new inhibitors had better interactions compared to their parent molecules. They formed hydrogen bonds with key amino acids like His263 and Ser152, and also had hydrophobic interactions with residues such as Phe77 and Tir114. One of the standout compounds from this research was compound 9, derived from the natural inhibitor vibrolactone. IC50 values showed that compound 9 had a remarkable inhibition activity against pancreatic lipase at 14 nm, that's over 3,000 times more potent than vibrolactone. In vivo studies demonstrated that this compound significantly reduced triglycerides, cholesterol, and high-fat diet-induced obesity in mice at a dose of 100 mg per kilogram. Next, we'll explore key figures from a recent paper, breaking down the concepts in a clearer and engaging way. Figure 1, QSAR Model Overview. First up, let's talk about Quantitative Structure-Activity Relationship, QSAR, models. These models help us predict the activity of chemical compounds based on their molecular structure. The study utilized the Monte Carlo method to build these models. The best model achieved impressive statistical parameters. R superscript 2 equals 0.864 and Q superscript 2 equals 0.836 for the test set. R superscript 2 equals 0.824 and Q superscript 2 equals 0.819 for the validation set. This means the model is quite reliable in predicting the activity of pancreatic lipase inhibitors. Figure 2, Descriptors Impact. Next, let's discuss the descriptors that were identified as significant for enhancing activity. Branching, the presence of branched structures improves interaction with the target. Oxygen with branching in double bonds, these features seem to enhance the compound's effectiveness. sp superscript 2 carbon and cyclic rings, these structural elements also positively impact the activity. Understanding these descriptors helps researchers design better inhibitors. Figure 3, Novel Inhibitors Design. Now, let's look at how these QSAR models were applied to design novel pancreatic lipase inhibitors. The study successfully designed new compounds with improved predicted activities, as shown in Figure 9. Molecular docking simulations were conducted to assess how well these new compounds interact with the enzyme. Figure 4, Docking Results. Moving on to molecular docking simulations, which provide insights into how compounds bind to pancreatic lipase. Figure 11 illustrates the docking of phenolic compounds like quercetin, picoumaric acid, and caffeic acid. These compounds demonstrated better interactions compared to their parent molecules, forming crucial hydrogen bonds and hydrophobic interactions with key amino acids in the enzyme. This highlights the importance of structural modifications in enhancing inhibitor potency. Figure 5, lead optimization. Next, let's discuss lead optimization using computer-aided drug design, CADD. Vibrolactone, a natural beta-lactone PL inhibitor, was structurally modified to enhance its activity. The optimization process resulted in a series of 104 analogs, with compound 9 showing remarkable inhibition activity, IC5 equals 14 nm, significantly more potent than the original vibrolactone. This demonstrates how computational techniques can streamline the drug discovery process. Figure 6, in vitro and in vivo results. Finally, let's touch on the biological evaluations. In vivo studies showed that compound 9 significantly reduced triglycerides and cholesterol levels in mice, showcasing its potential as an anti-obesity agent. In vitro studies revealed that compounds like quercetin exhibited mixed-type inhibition against pancreatic lipase, with varying IC5 values. These findings underscore the therapeutic potential of these novel inhibitors. To wrap up, this research highlights the power of computational methods in drug discovery, especially in the fight against obesity. By utilizing QSAR models and molecular docking, scientists can design more effective pancreatic lipase inhibitors, potentially leading to new treatments for obesity. If you found this video informative, don't forget to like and subscribe for more content on cutting-edge research. Thanks for watching, and see you next time.
Pharmapal.